discussing here steroids, human steroids, and we had discussed in previous part, we had discussed about what are the steroids and what are the structure of steroids, and we had learned there steroids are the cyclic molecules which contain 4 rings. Out of 4 rings, 3 rings are cyclohexane ring, and that cyclohexane ring is attached with a cyclopentane ring. So totally we can call it that along with 3 rings plus that one 5-membered ring we called is steroid nucleus. That steroid nucleus, all the human steroids, all the human hormones, or we can say that steroids which are present in our physiological system contain that steroid nucleus. And we had learned there different type of steroid, corticosteroid, glucosteroids, estrone, progesterone, testosterones. Now you are looking bile salt. This is the bile salt which is present in our human body, and cholic acid here secreted from linear, and it is a 5-membered side chain. At C17 position you can see there is a side chain, that is CH2, CH2, COH, right? So this side chain end with COOH group, carboxyl group. So that is called cholic acid. So learners, this is the bile salt, and as we know bile salts are very important part of human biological system. So that is why we called it cholic acid, because it is having a acidic group, COOH group. Along the COH group there is also 2OH group which is attached to a ring A and ring C, and there is also 2-angular methyl group which is present at C10 and C13 position. So total number of OH is over here 3. One is attached with ring A, one is attached with ring B, and another OH group is attached with C as you are looking on your screen, and there is a side chain which ends with COOH group, that is the carboxyl group. Learners, as I told you, all the sex hormones like androgens, estrogens, these contains ketonic group, hydroxyl group or acetyl group at C17 positions. So these are also steroids. So you can say this is structure of testosterone, and this is also testosterone, and it's having a androgen nucleus, and this testosterone is responsible for the male characteristics. So this is the structure of testosterone. Again, as I told you, it is steroid and it contains 4 ring. 4 ring out of 4, 3 ring A, B, C is a cyclohexane ring and D is a cyclopentane ring, and that cyclopentane ring, total number of the steroid nucleus is same in all the steroids, in all the hormones. But here you can see in testosterone, the cyclopentane ring having one OH group, one 2CH3 group, 2CH3 group present, that steroid nucleus, and ring A having carbonyl group, that is ketonic group. So the testosterone hormones which is responsible for the male characteristics having ketonic group. This is the progesterone. It is a female hormone and responsible for the characteristics of female. And you can see here in testosterone, the D ring, that is the cyclopentane ring, contain OH group, but here there is 2 ketonic group. The D ring having COCH3 group, methyl ketone group, and the ring A having ketone group, CO group in the which is attached to the ring A. So it having COCH3 group. Similarly, steroids or steroidal alcohol are amphipathic lipids. Why they are amphipathic lipids? Because they are having OH group at C3 positions in ring A in the sterol moiety, and also these hormones contain side chain at C17 carbon atom, having total number of steroid nucleus having C17 carbon atom, and that steroidal nucleus contain a side chain. That side chain contain 8 to number of carbon atoms. According to that, there are 3 types of sterols. That is, sterols means the steroid nucleus which contain OH group, right? So according to that, there are 3 types. Zoosterols. These zoosterols are obtained from animals or found in animals kingdom. Very important example, and we all are aware of that, cholesterol. So cholesterol is a zoosterols, and another example is coprostenol. Another types of sterol is which are having OH group, that is phytosterols. Phytosterols are obtained from the plants. An example of phytosterol is stigmasterol and cetosterols. And learners, by consuming of that phytosterols which are present in plants, we can decrease the high level of cholesterol in human system. Another type of sterol is mycosterols. So microsterols which are present in yeast and fungi. Example of mycosterol is ergosterols and mycosterols. So all these are the 3 type of sterols: zoosterols, phytosterols and mycosterols, which contain OH group in the steroid nucleus. This is the structure of mycosteroid. So this mycosterol, again learners, as I told you, all the steroids having same steroid nucleus. So this mycosterol contain OH group at the ring at carbon atom, at 3 carbon atom, and also the cyclopentane ring, which is a D ring on your screen, you can see, is contain a long chain of carbon, CH3, CH2, double bond, CH, then CH3 having CH3 over that carbon atom, CH, CH3 and then CH3. So this is the mycosterols, and this found in yeast, and it is a precursor to vitamin D. Now this is a structure of stigmasterol. As I told you, it is a plant sterols, and you can see here the OH group is present in ring A at C3 carbon atoms. So this sterol, which is a plant sterol, we can say this is a plant sterol, and this is found in fats, oil of soybean, seeds, almonds, nuts. So this is a stigmasterol. Now very important part of this lecture is cholesterol, which is found in our body. What is the cholesterol, learners? We are all aware of this term, cholesterol. So learners, cholesterol is also a steroid. It is also human steroids present in our human body. Now this is the structure of cholesterol. As I told you, it is also having a steroid nucleus, that steroid nucleus contain 4 ring. Now it is also type of all zoosterols, as in the previous slide you have seen, it is a zoosterols found in animal kingdom. So this steroid having OH group at C3 carbon atom. Along with that, very important part is a long chain of carbon present at C17 position in cyclopentane ring. So you just remember steroidal nucleus and C17 carbon, there is that carbon chain. So it is cholesterol is most abundant member of steroid family. It is also called blood fat. So it runs in our blood along with the protein, that is called lipoproteins. So it is also a class of lipid. And learners, very important, it is precursor to all the steroid hormones, bile acids and vitamin D. How cholesterol looks? Cholesterol is a waxy fat like substance and it present in all cells in our body. And it's very important, learners, our body make all the cholesterol it needs. So liver, especially liver, can make 80% of cholesterol which we require, and can be found in food from animals at 20%, egg yolk, meat and cheese. So out of 100%, the 80% of cholesterol is synthesized or biosynthesized by liver in our physiological system. This cholesterol is very important, play very important role in regulating cell functions. Cholesterol is amphiphilic liquid molecule and it intercalate between phospholipids in the lipid layer. Phospholipids is a lipid which contain alcohol molecule and lipids molecules, phospholipids especially, phosphate groups. If we talk about the structure of, it's very important fact and you have to remember, learners, we have seen the structure of cholesterol. There is a 8 center of chiral center. So according to the chirality, there should be 256 stereoisomers possible for the cholesterol, but in nature only one type of isomers exist. On your screen, the red dots which I have represented here represent the chiral center. Chiral center, what are the chiral center? The carbon which is attached with 4 different groups, right? So the total chiral center in cholesterol is 8. So according to the rule, the 2n square, here the number of stereoisomers should be 256. But there is only one stereoisomers is present, that is the cholesterol. What is the reason? It's very important. During the biosynthesis, all forms of isomers come together and to form a type of steroid, that is Lanosterols. We will discuss in the biosynthesis of cholesterol, but you have to understand the reason here. When the biosynthesis takes place in our human system, biochemical reaction takes place, all form of isomers come together and to form only one form, that is Lanosterols, and that is a ring closing steps, and that Lanosterols has 7 centers, 7 chiral centers. All of them are fixed by Lanosterosynthase enzyme and we get only one enantiomer 8 times, right? So during the biosynthesis we get a Lanosterols because the biosynthesis of a cholesterol is cascade of enzymatic reactions. during the biosynthesis of cholesterol, a lot of enzymatic reaction takes place, and during that enzymatic reactions we get a lanosterol, right, and the lanosterols having 7 chiral carbon atom, and in presence of that lanosterosynthase enzyme we get only one isomers 8 types. So that is why, that is the reason only one stereoisomers, which is the cholesterol, exists in the nature, and the molecular formula of cholesterol is C27H26O. It is a odorless and tasteless molecules. Now come to the important part, how the cholesterol is biosynthesized. It's very important. First part is the cholesterol is biosynthesized by the mevalonate pathway. As I told you in the previous lectures, during the mevalonate pathway we can biosynthesize isoprenoid units, and that isoprenoid unit condensation of takes place and to form a squalene. Learners, squalene is a very important molecule. It is a key molecule for the biosynthesis of all the steroids hormones. And how can we biosynthesize the squalene? It is biosynthesized for the terpenes, and when this squalene molecule is biosynthesized, cyclization takes place, and during that cyclization, during that reaction we get lanosterols, and from the lanosterols we get cholesterol. So lanosterol to cholesterol, it is a multi-step process. Almost formation of biosynthesis of cholesterol from lanosterols required 19 steps. So that's why I have called you, I have told you here, a cascade of enzymatic reaction takes place during the biosynthesis of cholesterol. It's very important, which I have told you, squalene. So this is the structure of squalene. Squalene is an intermediate in the biosynthesis of cholesterol, and it is a terpene. Here, how many terpenes unit, isoprene unit is here? 8 isoprene unit, and on the structure you can see here that squalene is saturated hydrocarbon compound, a long chain of saturated hydrocarbons, and 8 isoprene unit. It is biosynthesized by the coupling of 2 molecules of farnesyl pyrophosphate and used to synthesize terpenoids, terpenes and sterols. So this is the molecule of farnesyl pyrophosphate. I will here represent here GPP. So 2 molecule of GPP and 1 molecule of GPP and isoprenoid pyrophosphate, we get GPP. So it is the method, it is a biosynthesis of geranyl pyrophosphate, and geranyl pyrophosphate is very precursor to squalene. So it is a very important intermediate for the biosynthesis of squalene. That geranyl pyrophosphate and 3-isopentyl pyrophosphate react with each other to form farnesyl pyrophosphate. So this is the structure of farnesyl pyrophosphate. That farnesyl pyrophosphate on your screen, this is a terpene. Learners, this is a terpenes, and this farnesyl pyrophosphate contain 3-isoprene unit along with phosphate group. Now in the first step, after biosynthesis, after the formation of squalene, the conversion of lanosterol takes place. And how the conversion of lanosterol takes place? Epoxidation takes place. Epoxidation of 2,3 double bonds of squalene takes place. After that, acid catalyzed opening of epoxide ring which initiate a series of cyclizations to form protosterol cations. Now, after the formation of protosterol cation, elimination of a proton from the C19 carbon atom which initiates a series of 1,2 hydride and 1,2 methyl shifting, because you have seen in the squalene unit there are a lot of methyl group. So, after the elimination of C9 proton, 1,2 hydride and 1,3 methyl shifts takes place and we get a cyclic structure, that is Lanosterols. So, this is the reaction on your screen. You can see farnesyl pyrophosphate in presence of enzymes convert into squalene. Then that after the squalene formation, converting of Lanosterol to cholesterol, it requires 3-methyl group removal. So, always remember, conversion of Lanosterol to cholesterol, we need to removal of methyl group, and for the removal of 3-methyl group, we need at least 19 steps. So, the reaction takes place a very long pathway, there is a very long pathway, and after 19 steps, when the all 3-methyl group removed, we get cholesterol. So, you can see on your screen here from squalene due to a lot of enzymatic reactions, and finally Lanosterol convert to cholesterol. So, as I told you that Lanosterol contain also chiral carbon atom, but during the enzymatic process all the isomers convert into only one form, that is cholesterols. It is very important, learners. Why does nature bother, and why not just use of Lanosterol instead of cholesterol? Have you think about it? The reason is that it was found that membrane that having Lanosterol is more permeable. Even small molecules are able to pass easily through the Lanosterol containing molecule. As each 3-methyl group removed from the Lanosterol, the membrane become less and less permeable. So, permeability decreases as the removal of methyl group takes place from the Lanosterol. So, this is the reason we are having cholesterol in our biological system. On your screen, that is the structure of vitamin D3. You all are aware of vitamin D3. Our skin can synthesize also vitamin D3, and or we can take vitamin D3 from our diet. So, here vitamin D3 during when we take the sunlight, here that calciferol on hydroxylation in presence of 25 alpha hydroxylase convert into calcidiol. So, this vitamin D is also converted into a very important molecule, that is calcidiol, which is present in our liver. And learner, when calcidiol is increases in the liver, what happen? That calcidiol, your structure looking on your screen, that calcidiol in presence of 1-alpha hydroxylase enzyme converted into calcitriol in kidney. So, you can see a series of biosynthesis reaction takes place in human physiological system, and that calcidiol, which is from form of vitamin D, produced in the liver by hydroxylation. So this is the structure of calcidiol. So here A ring having OH and the side chain is also having OH. It is an active form of vitamin D. Learner, deficiency or excess of these form of cholesterol, because these are all are form of cholesterol. So when deficiency or excess of cholesterol takes place, we can get a gallstones. So calcitriol is responsible for the gallstone, because in this case whether that cholesterol or calcidiol is in excess in kidney or in deficient amount in kidney or in bile salt, we get a gallstone. So this is the reason why stones can takes place in our kidney. It's very important. Synthetic steroid. What are synthetic steroids? Learners, synthetic steroids are man-made version of steroids, and they are also called anabolic steroids or synthetic steroids. These steroids used to aid in the development of muscles. The muscles building, which is affect as a testosterone deficiency or takes place. When the deficiency of testosterone takes place, the building body muscles can be affected. So these are the anabolic steroid, man-made steroid, sometimes taken by persons. So these human-made steroid or anabolic steroid available only by prescription and used to treat suffering from traumas accompanied by muscles deformations. They are also called anabolic androgenic steroid, ASS. They are structurally related and similar to effects of the testosterone. They increase protein within the cell, especially in skeletal muscles, and have varying degree of virilizing effects, including induction of development, maintenance of masculine secondary sexual characteristics. And it's very important, their use is referred to as a doping and is banned by most major sporting bodies. And there are long-term effects of these steroids, these anabolic steroids. It affects on our kidney, liver and heart swelling, heart failure, hair loss, high blood pressure, high cholesterol. And learners, sometime same drugs have been administered to athletes and race horses to increase muscle mass, and the majority of people who misuse steroids are weightlifters and also detected in several athletes. So these are the some anabolic steroids or human-made steroids. Their structure is resemble to the natural steroids. RU486, these used to treat symptoms of Cushing syndrome and pregnancy termination, and they enhance the steroid doping. They are the steroid doping substances. These are the structure of these steroids. It's very important point, learners. Cholesterol and other fats carried in blood as a spherical particle, that is lipoproteins, and there are two types of lipoproteins, that is LDL, which is low density lipoprotein, which is also bad cholesterol, and that bad cholesterol is contribute to the formation of plaque build up in the arteries that linked to higher risk of heart attack and stroke. So in this part we had discussed about the biosynthesis of squalene and how biosynthesis that squalene can be converted into cholesterol. So we had discussed about the biosynthesis of cholesterol in detail, how the that squalene after the epoxidation reaction and lot of enzymatic reactions convert into Lanosterol, and Lanosterol convert after the 19 step change into the biosynthesis of cholesterol takes place. Thank you.